Bismillahirrahmanirrahim. We start the topic-wise MCQs. This is chapter 2, biological molecules, and we will discuss the MCQs on this chapter. Starting with question number 41, because video 1 had questions 1 to 40, so this is the second video. Which row is correct when one molecule of sucrose is hydrolyzed? Molecules of reducing sugar 2. Why? Because sucrose is made up of a glucose and a fructose. So there are two monosaccharides joined together by a 1,2 glycosidic bond. And then molecules of water released is 1. So this was the question 41. Then 42. The formation of glycosidic and peptide bonds is responsible for polymerization in some biological molecules. Which bonds are formed in each molecule? So in glycosidic, in glycogen, and peptide in glycoprotein. So I mean they've just tried to confuse you because the glycogen part is the one which is, you have to remember, is just a glycogen carbohydrate part which joins up with the protein part. So the answer to that was and here, of course, glycerol is wrong because glycerol is found in fatty acids and glycerol make the triglycerides. So just to confuse you, the glycogen and glycerol look very similar. So that's where students are going to get caught up and make a mistake. Then the diagrams show the structures of two amino acids, each of which has more than one amine group. A peptide bond is formed between the two amino acids. Which groups form the peptide bond? Which groups form the peptide bond? Is it 1 and 4? Is it 2 and 4, 2 and 5, 3 and 4? So the answer was, it was 1 and 4. You know there's N and C joins, because this is what we have to understand, is because the amino acids form a peptide bond. Now, as you can see here, this is a peptide bond. So the peptide bond is, where is it, so we've got to understand where the peptide bond is formed. So this is one amino acid, right? And this is the second amino acid. So the N and then the central carbon and then another carbon, and this is the N. And so it is between the N and the C that this is the peptide bond. So this is just a quick revision of what we have to understand. And a molecule of water exits, and this molecule of water which exits, is the H comes from the N side and the OH comes from the carboxylic acid side. So the water molecule will exit, and this, of course, then that is why is called a condensation reaction. And if you add water, it will break this bond. If you add water, the water will split into H and an OH, and it will sit, the H will come here and the OH will come here. So this is what you have to understand, the OH coming in here and the OH, and of course this bond breaking, and that will be hydrolysis. Hydrolysis means the breaking up of a water molecule into hydrogen and OH and H ion. And question 44, which explains why hemoglobin is able to carry oxygen? It contains iron. The iron attaches to Fe2 plus, the oxygen attaches to the iron part of the hemoglobin molecule. The 45, the R groups of side chains of amino acids can be hydrophobic, hydrophilic, acidic, or basic. And the diagram shows 4 different amino acid R groups. Which row is correct for these amino acid R groups? So hydrophobic, hydrophilic, acidic, basic. Well, you have to figure this out because you can easily tell which is which. And of course, you need to revise it from the book as well to really be understanding this. Then coming on to question number 46, which describes the emulsion test for the presence of liquid. Add ethanol, pour water and shake. Add ethanol and shake was wrong. Add water and shake was wrong. Add water, pour into ethanol. No, you have to add the ethanol first and then the water later on, because if there's fat, it dissolves in the ethanol. Then question 47, the structural form of carbohydrate molecule can be shown as this. Which of the molecules could be represented by the formula? Now, because this is a 1, 2, 3, 4, 5, 6 carbon, so it cannot be ribose. And sucrose is a disaccharide, so it's one only. Question 48, the diagram shows part of a molecule of glycogen. You know glycogen contains 1, 4 and 1, 6 bonds. How does this molecule, glycogen, differ from a molecule of amylase? Amylose. Amylose only has 1, 4 glycosidic bonds. So you can see where I want to mark these, and I want you all to see this. This is where there's 1, 6. This is where there's 1, 6. And wherever it sort of, branches off, that is where it's 1, 6. This is a 1, 6. Uh, This is a 1,6. This is a 1,6. And then this is a 1,6 here. Then this is a 1,6 here. Then a 1,6 here. Then let me see if I've missed out any. This is a 1,6 here. So you see where the straight chains are present. You can see this is the straight chain. And then this is the straight chain. And then this is the straight chain. And then this would be the here where it branching here, this would be. And then we have the straight chain here. This one, of course, is a straight chain. And then this one is a straight chain. So the ones in blue would all be 1,4 glycosidic bonds. And these would all be the 1,4 glycosidic bonds. So it's what here, then this would be a 1,4, then this would be 1,4, then this would be 1,4, then this would be 1,4. And so please understand this, wherever I put the red mark, that would be the one, that would be the 1,6. So the one here, this would be the 1,6 glycosidic bond, and the blue ones would all be the 1,4 glycosidic bonds. So that was the difference, and everybody needs to revise this and be very clear on this. Then question number 49. Which molecules contain a carboxyl group? So amino acid, and it was amino acid 1, 3, and 4. So 1, 3, and 4. Glycerol does not have a carboxyl group. Then question number 50. The diameter of some atoms when they form bonds are given in the table. The appropriate length of the amino acid shown was estimated during using the figures in the table. So we need to do this, and then it says what would be the approximate length of a dipeptide formed using this amino acid. Dipeptide means that there are two amino acids and there is a peptide bond between them. So there's one amino acid and the second, like you have disaccharide. So dipeptide doesn't mean two peptide bonds. It actually means two amino acids, but one peptide bond. So there's one peptide bond. Now, what would be the length of this? Now, I'm going to just do the calculations and show it to you. Now, as you look at question number 50, I estimated it like 2H are removed, so 0.06 twice, this and this. And then O also was removed because we are removing this O, this H, and this H. And then we form a bond between N and C. And then O is 0.132, so we added it 0.25. And because the total, if we have two of these, you see there's one peptide, one amino acid, and then we have another amino acid, so they both will be 0.7 plus 0.7, that is 1.4, and 1.4 minus 0.2 is 1.2. So I think that is how we have to figure this out. Maybe you can come up with another way as well. I mean, I'm open to suggestions. 51. Which feature of water is least likely to affect the life of an animal in tropical rainforest? Maximum density at 4, that's only when ice freezes and then it starts to come to the surface, and of course that means the fish in the bottom layers will survive. This has also been removed from the syllabus. Then two pairs of students each counted the number of bubbles of oxygen given off in a minute when investigating the effect of catalase on plant tissue on hydrogen peroxide. Each pair repeated the experiment 5 times, calculated the mean, which correctly calculated the mean. But of course, because you see, bubbles cannot be in fractions. So the fractions were all wrong. You can't have zero point 0.2 bubbles or 0.1 bubble or zero point 0.8 bubbles. That was not possible. So the only answer was 2, and that was just a little catch in the question, which somebody had to figure out that the fraction was wrong. You can't count the bubbles in fractions. Then question 53, heating with which solution breaks glycosidic bonds? Yes, dilute hydrochloric acid will break the glycosidic bonds between the glucose and fructose. Then the diagram shows four molecules. Which two molecules condense to form sucrose? And you know sucrose is between glucose and so P and S. This is a fructose and forms a 1,2 glycosidic bond. I'll just show you a picture of it. You can revise this molecule of sucrose here, and it is made up of a glucose molecule and a fructose molecule. And you see this is a alpha 1,2 glycosidic bond. So fructose is actually a glucose isomer, but still, of course, its structure is slightly different, but it is the same C6H12O6, so alpha 1,2 glycosidic bond. Please revise this, and this is, of course, part of your syllabus now in the new syllabus as well. So 54, you can see the diagram shows four molecules, which two molecules form sucrose. So the answer was P and S and glucose, and we've just revised that. I hope that is clear to you. Please make a mental note of that and remember it. Now, which molecules have a structured formula that contains C double bond O? Now we know that proteins contain that C double bond O and glucose contains it. So this is what we have to understand is 1 and 3 only. Then question 56, hemoglobin consists of 2 alpha, 2 beta. Approximately 5% of all humans have one amino acid in the beta chain different from normal. Which levels of protein structure could be changed in these? So of course, if it's the primary structure, then the whole secondary and tertiary will also change. So primary, secondary, and tertiary will all change. Which of the statements about polysaccharides can be used to describe both glycogen and amylopectin? So contains 1,4, contains 1,6, and polymer of alpha glucose. Yes, so it's all three of them. They were 1, 2, and 3 because yes, alpha glucose hai, 1,6 bhi hai, and 1,4 bhi hai. So it's both glycogen and amylopectin both have 1,4 and 1,6. Glycogen is more frequently branched. Then we have question 58. Which row shows the type of bond found at different levels of protein structure? So peptide bond is in the primary, P for peptide and P for primary. This is how you remember it. And levels of protein structure bonds, right? Now, which one was correct? You have to see which line was correct. So tertiary contains hydrogen ionic and hydrostatic, because primary does not contain hydrostatic. So this was wrong because this was wrong. Hydrostatic was wrong. And secondary was wrong because it only contains hydrogen, not ionic. So this was wrong. Tertiary was correct. C was correct. D was wrong because peptide bonds would be in the primary structure, not in the quaternary structure. So this was wrong. And also ionic will not be present, and also not hydrostatic. It would be the bonds between this is usually hydrogen bonds only. A student carried out a series of tests and an extract from a plant. The table shows the results. Ethanol and water white emulsion, so fat present. Benedict's brick red precipitate, so reducing sugar present. Biuret blue color, so no protein in it. So protein was missing, so it either has to be B, C, and D. Fatty acids were present, yes. And reducing sugar was present, yes. So the answer was B. Then coming to question number 60, which glycogen is a polymer of glucose. Which description summarizes its structure? Alpha glucose joined by glycosidic bond involving carbons 1, 4, and 6. So that is how you have to describe glycogen. Then which molecules contain C double bond O, 1, 2, and 3? This is, of course, we've done this before. 62. Which statements about polysaccharides can be used to describe both amylose and amylopectin? Contains 1, 4, 1, 6 polymer, so 1 and 3 only, because amylopectin, it says amylose and amylopectin. So the common thing in that is 1, 4, and it's a polymer of glucose. So 1 and 3 were the answers to this. Question 63. Which carbohydrate gives a brick red color when heated with Benedict solution? Glycogen, no. Sucrose disaccharide, no. Cellulose, no. So it's only fructose that was the answer. Then 64. Which diagram shows, the diagram shows two molecules of glucose. Four possible bonding positions are labeled P, Q, R, and S, and T, U, V, W. When these two molecules contest during the formation of a glycogen molecule, where would bonds form? So P, U, P and U. So P is here, and where is U? U is here. That would be the 1, 6. P and U, and Q and W. Q is here and W is here. So Q and W. So there, of course, this would be the 1, 4, and water would exit. And this would be the 1, 4, and this would be, of course, the 1, 6. So this is the 6, so it had to be P and U, which is the carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Similarly here 1, 2, 3, 4, 5, and carbon 6. High concentrations of urea break all bonds except covalent in the protein molecule. Which levels of protein structure would remain unchanged when a protein is treated with urea? Covalent bonds are the peptide bonds and the disulfide bonds, but other bonds, of course, are not. So, covalent primary would, of course, remain intact because the peptide bonds are covalent bonds. Then, two disaccharides are cellobiose and sucrose. Cellobiose is formed from two molecules of glucose, while sucrose is formed from fructose and glucose. Which row shows the molecular formula of the two disaccharides? And I've done this in a previous, the first video as well. It's C6H12O6 plus another C6H12O6. So we get a total of C12, H24, and O12. And from this you minus H2O. So this will become 22 and this will become 11. So C12, H22, O11, and this has come in a previous question as well. We've done this before. The diagram shows part of a carbohydrate molecule. You can see here this is the 1,6 bond, and these are all 1,4 bonds. So 1,4 and 1,6 are present in amylopectin and glycogen and starch. So amylase only has 1,4. So 2 can't be in it, so this would be wrong, this would be wrong. And 3 and 4 is not enough. 3 was glycogen and 4, but we had to have 1 as well. So this was the answer to question 67. Then coming to 68. Solutions of biological molecules are tested for sugars. The table shows the colors of the solution after testing. One is blue to orange. So blue means no reducing sugar. Orange means non-reducing sugar. Orange and green. So this had more of the reducing sugar, and then less. The sucrose was present, but it was much lesser than this one. The 2-1 yellow and then red. So this yellow means slight reducing sugar and red means a lot of non-reducing sugar. So which may contain non-reducing sugar is 1 and 3. '69. When a lake begins to freeze, which properties of water needed for the fish to survive? Water has a high surface tension, water has a high latency of vaporization, water has a high thermal capacity, and water has its maximum density at 4 degrees Celsius. So number 3 and 4 were correct. High thermal capacity, and I think this has been removed from the syllabus. Then question 70, which polysaccharides are branched and which are unbranched? So amylopectin is branched, glycogen is branched, cellulose is not branched. So the only answer was this. Then 71, three carbohydrate molecules are shown. Which two molecules to combine to form a molecule of sucrose? So it is 1 and 3. At which level of protein structure do ionic bonds occur? Ionic bonds occur in the they occur in the tertiary and the quaternary. So it was 2 and 3. They would occur. Myoglobin is a protein with similar function to hemoglobin. However, myoglobin does not have a quaternary structure. Why does myoglobin not have a quaternary structure? Because myoglobin has only one polypeptide chain. Quaternary is always when there are two or more polypeptide chains. The diagram shows the structure of the amino acid cysteine when two of these amino acids join together. So there's another one joining here. So if we have another NCC here, then this is an H, and then it's an H. So this H and OH will leave, and between this C and N, a peptide bond will form. And so this is the peptide bonds only. Which row describes a triglyceride, hydrophilic, and insoluble in alcohol? Yes, question 75 was slightly, I misread it. Triglycerides are fats and they are soluble in alcohol. So that is why it's wrong to say they're insoluble in alcohol. And they are not hydrophilic, so that's why the answer was D. Then the diagram shows part of a carbohydrate molecule. And then what does it say? If all the 1,4 glycosidic bonds are hydrolyzed, how many water molecules will be used and how many separate glucose molecules will be produced? Now that was it wasn't very difficult, but of course if you don't understand it, then you are going to struggle with it. Now you see why the answer was this. Now you can see why I've made the yellow are the two glucose molecules. So the number of glucose molecules produced will be these, the two with the yellow ones. Now you realize that this is still a disaccharide. This is not going to break because it doesn't break the 1,6. This is the 1,6 bond. That is not going to break. So it said if all the 1,4 are hydrolyzed, how many water molecules will be needed? So 2 will be needed. And how many separate glucose molecules will be produced? So only 2 will be produced. The two which I have drawn in yellow. This is one, and the other one is this one here, which I have drawn in yellow. A peptide bond is formed between atoms of which two elements? It's carbon and nitrogen. I've just shown you the peptide bond in the earlier diagram. Which features affects the tensile strength of collagen? It's 1, 2, and 4. The helical structure of collagen, the small R groups of the amino acids in collagen, and the covalent bonds between the collagen molecules. So the strength is because of these three things, 1, 2, and 4. Not the insoluble nature of collagen. That is not the reason why it's so strong. An amino acid can be represented as where R groups are a variable side chain, which is not a possible side chain. So the answer to that was D, that is not a possible side chain. 80. When red cells leave the lungs, hemoglobin is 98% saturated. How many molecules of oxygen will be carried by the majority of hemoglobin molecules? 4. 8 atoms. 4 molecules, 8 atoms. 81. Which feature of hemoglobin makes it a globular protein? The hydrophobic R groups are on the inside and the hydrophilic groups are on the outside. A student tested 4 samples of food A, B, C, and D for the presence of lipids, reducing sugar, proteins, and starch. One of the food sample, milk, was found to contain lipid, protein, and reducing sugar. Which of the food samples shown in the result below is milk? So if it is lilac or purple, then protein is present. So this is the protein part that was present. And then if it is orange with potassium iodide, then that means it does not contain any starch. And boiling with Benedict's orange precipitate, that means reducing sugar is present and it forms a thick milky white emulsion. So you see, this means starch is present. Now this would be wrong because we don't want any starch in it. We just want lipid, protein, and reducing sugar. And why is the D wrong? Because it gives blue, so that means this does not have any reducing sugar. So this was wrong. Then question 83. Which diagram represents part of the ring form of a molecule of glucose? Now that was A. Why are the others wrong? You've got to figure it out. Why are they wrong? Part of the ring form, you see, this is what you have to understand. The ring form of the molecule of beta glucose. Yes, the beta glucose was the important part. Which statement describes how the molecular structure of starch is suited to its function? The final amylose amylopectin complex is insoluble and does not affect the osmotic properties of the cellular. I told you CIE, compact, insoluble, easily hydrolyzed. Compact, insoluble, easily hydrolyzed. That's what storage molecules should be like. Which feature distinguishes starch from glycogen? Starch has an unbranched component. Glycogen is all branched. Starch has a component. Part of it is amylose, part of it is amylopectin. Amylose does not branch. So that is why that is the difference between glycogen and starch. This is the marking key for this file, and you can pause it here, and if you want to go through it again, then you can just revise it. This is the video 2 for the biological molecules, and I hope now this is helpful to all of the students taking the October-November exam for A-level biology.